Hello, people. What do we got? We got 22 people. How many are in the class? I should know this, right? I'm supposed to be in charge. Let's see your faces real quick so we can pretend we're a classroom. And hey, Mairead, Nishi Kenderwall, I'm going to answer your email shortly. Elijah Cummings. Rocky Raccoon. There she is. Christopher James, still not smiling. You know you can. Come on, buddy. There's Rocky. What are you eating, Rocky? It's lunchtime. You can't eat in front of us.
I'm eating sushi.
Oh. Okay. All right, what do we got now? We're up to 24. That's pretty good. So today we're just doing a review. I'm going to pull up the PowerPoint and I'm going to tell you as we go through the PowerPoint what I said in the first place, I hope. And then you could ask me questions on what's going on. I'll hopefully tell you, can you please give everyone 100 on the test? Yusuf, I like your ambition. Ain't going to happen though. I only give that to Nishi. Nishi, you don't have to take the exam, don't worry. What the hell was I talking about? Oh, and Tyler, I forgot Tyler. Yeah, you get an A too, because it's your birthday soon, Yusuf. I have a birthday too. What'd you guys give me for my birthday? Exactly. Okay, so what the hell was I talking about? All right, so I'll go through this PowerPoint. Stop me anytime with a question. Hopefully, as we go through, I'll point out what's most important, what to keep in oh, Vanessa's got the dog. Hey, puppy. I hate to tell you, but my big boy would eat that dog for breakfast. Look at it, Gus. Look at. Oh, you can't see him. The virtual background. So I know this is going to shock you all. You're going to be shocked when I say this, but when I was going through the PowerPoint today, I found that I had made some mistakes. So I corrected those mistakes, and I will delete the PowerPoint that's in your Moodle page and put the new PowerPoint back up. Also, one of the mistakes I made was somewhere around lecture 4, I forgot that veg tea was called veg tea and started calling it veg 1. So I corrected that in your notes. So I'll put your I'll update that as well in your notes, in your Moodle page. Okay, with no more ado, go to our PowerPoint. Come on, baby. Okay. Again, don't be afraid to stop me if you've got a question. I can't keep my chat box open. So Tyler, I can see you there on my screen. So if someone says something in the chat box, interrupt me and tell me to read the chat, okay? Because I can't really see it.
Okay.
Okay. All right, Xenopus fate mapping. Now, one of the things that, this is the third time I'm doing this class, so I'm starting to figure the textbook out a little better. And one of the things I find really annoying about this textbook is it like says, okay, this is what causes development. Now let's go back and talk about how that was found out through these experiments. Now let's go forward and do this. Now let's go back and do that, and forward and do this, and back and do that. And I think the next time I teach this class, I'm going to rearrange the whole thing differently because it's kind of hard to follow. But anyway, this is our bottom line. This is what we're going to try and learn through this chapter is like, why do these tissues become these tissues and not other ones? Because, at this blastula stage, this isn't blood, kidney, and heart mesoderm, right? This is just generic mesoderm, ventral mesoderm, really. So inductions happen along the way and cell fates change. And we start out with our egg cell, animal, animal pole, vegetal pole, and we've got maternally laid messenger RNAs here that are going to cause our dorsal lip to form, our Spearman organizer. Keep in mind too that it's important that they're messenger RNAs because you're not getting any transcription from the zygotic nuclei, right? You're not making any messenger RNA from the cells themselves. So the only proteins being made are the ones that are made from messenger RNAs placed in the egg originally. You can thank mom for that. Okay, so after fertilization, you get one split of the end of the embryo, and now you've got two different cells. You can separate these two different cells, and if you do, because you have the dorsal determinants going into either of the cells, you get two complete embryos. A little smaller than the first one cell division less, makes a big difference at this stage. But they've got all the structures they need to survive. Now, if you wait till the second division, now you've got really two different types of cells because your dorsal determinants are here. So this only has ventral determinants. So here are your dorsal determinants. Here is just your ventral part. So if you take this ventral two cells and you put that in tissue culture, all you get is like a one-axis ventral tissue, really just a blob of nothing. The dorsal half, if you take though, because you've got the dorsalization factors here, that will form a Spearman organizer, and the Spearman organizer will give you axes. So because if you take this and put it into cell culture, you get what looks like a normal embryo. You've got anterior, posterior, dorsal, ventral, right, left. You've got all the three axes you want. Problem is you don't have a lot of gut here. I'd be glad to get rid of some of my gut right now. How many people put on the COVID-19? I did. Too much comfort food. Okay, so the thing is, if you've got these dorsal ventral halves here, then you will get the axes, but without the endoderm. If you take this half, you get only the ventral. So these are the early experiments they used to figure out that there's a difference between the different cells after the 4-cell stage. So there must be something inherent in these that give you that dorsalization. Again, this is before they have molecular determinants. Now at this point, you do your third cleavage, you've got 8 cells, and now basically you've got 2 different tissues, ectodermal tissue, well, really 4 different tissues. You've got ectodermal tissue, but this will be dorsal ectoderm probably. No, I'm wrong. Let me leave that out. You've got ectoderm and endoderm at this stage. All right. Now we talk about these dorsalizing factors. What are they and how do they get there? So this is the egg, and these things are here at the bottom of the egg. One is veg-T. Veg-T is not going to be moving. It's expressed all the way through this endoderm, and it's what makes this tissue endodermal. But here's some of the other factors that are attached to the cytoskeleton and will be moving up, attached to the cortex of the cytoskeleton. When the cortex rotates, they're going to move. What we've got here is, okay, so these are the endodermal things. VEG1, which is a TGF-beta hormone, VEG-T, which is a transcription factor, which is endodermal specific. And then you've got these dorsalizing things that are attached to the cytoskeleton and will move up into this area. Beta-catenin is the center of that. And we'll talk about these again shortly, so I'm not going to go into too much detail here. Okay, so there's your cortex, just showing you what the cortex is. And this is not an embryo, or I mean an egg cell. This is some kind of intestinal cell. But the point is the cortex surrounds the whole outside of the embryo. So it's all around here. So when the sperm enters, that cortex rotates, and these dorsalizing factors that were at the bottom come up to here. So your dorsal end of the, once the sperm comes in, that determines your dorsal ventral axis, because your dorsalizing factors will go opposite of where that sperm entered. Okay, what are those dorsalizing factors? They all involve beta-catenin. Okay, that's the key here, because you've got beta-catenin itself, but then you've also got 1, 2, 3 other things that are going to increase beta-catenin. Wnt is the hormone that will increase beta catenin. Disheveled is in the pathway, in the Wnt pathway, and it will also activate beta catenin production. And GSK3 binding protein, because GSK3 is in that Wnt pathway and it breaks down beta catenin, or helps to. And so all these things are leading to beta catenin. And then other things that are going to be released here and will diffuse away are, and these will be important for AP patterning, Dickoff, which is going to block Wnt signaling. I forget now whether it directly binds to Wnt or what it does, but I think it binds to Wnt. So this is going to be, it's ventrally located, so it's going to keep this Wnt signaling off in the ventral area. Okay, and here's our canonical Wnt pathway. And they call it canonical because it also has a G protein pathway that isn't used very often and not important to us, so we're not going to worry about it. But this is the main Wnt pathway. So in the absence of Wnt, there's no hormone out here. You've got these single transduction proteins that are just hanging around waiting for the signal to come. And that includes disheveled, frizzled, and LRP. We'll see this guy later. We've got a protein that blocks LRP, and that will block Wnt production as well, or Wnt transmission as well. And okay, so here is the death complex, or the destruction complex, I'm sorry, which is GSK3 beta, axin, APC, and CK1. So these are holding, basically these proteins are holding GSK3 beta in such a position that it can phosphorylate beta catenin. And when beta catenin gets phosphorylated, it gets directed to go to the proteasome and be destroyed. So that's what's happening when Wnt signaling doesn't occur. And because that's happening, you've got a bunch of repressor proteins here that are going to block transcription from the Wnt activatable genes. When Wnt is around, then it forms a complex with disheveled and LRP, and all of that then attracts GSK3 beta, axin, and CK1, and that stops GSK3 beta activity. So because GSK3 beta activity is off, beta catenin builds up. The way you want to think about that is beta catenin is constantly being made at a constant rate, okay? Wnt shows up and it stops being broken down, and boom, its concentration increases really quickly because it's no longer being broken down. And that's kind of why, without a bit of a digression, but that's why it happens this way, because beta catenin can be increased a lot faster this way than if it had to be translated from the mRNA. Also, you're not getting any messenger RNAs being made at this stage either, right? This is all proteins that are being made from maternal messenger RNAs. So throughout this period, you've got maternal beta catenin messenger RNA making beta catenin, and its levels being kept low by GSK3 beta. When Wnt comes around, then beta catenin goes up, and then you activate all these genes that are turned on by beta catenin. Okay.
I have a question, sir.
Okay, go ahead.
It's about Wnt, right? So if there's no Wnt, there's no beta catenin. But would this just mean that there would be no axes forming, like no dorsal ventral axis forming? What would this mean for like the cell itself?
For the embryo itself, you mean?
Yeah, would it just like abort?
It would. Okay, so what would happen is you get when we're at the blastula stage. Okay, so you've got veg-T at the bottom, which is producing veg-1, and also BMP and the nodal relateds, okay? They're all going up and they're making those marginal cells into mesoderm. Now, if you don't have any Wnt or beta catenin, you're not going to get a Spielman organizer, and that's as far as you're going to get, okay? You won't get any axes, but you will get mesoderm around the middle, but it'll all be ventral mesoderm, no muscle or bone, just blood kind of cells. And so you'll have epidermis, middle mesoderm, and endoderm, and that's it. That's as far as the embryo will go. Okay? Now the Wnt signal is going to activate that produced a new coop center, which then turns on the Spielman organizer, which is then going to cause gastrulation and the other axes to happen. Okay, Tyler? Does that make sense?
Yeah, it did. Thanks.
Okay, so if you knocked out beta-catenin, it would be just like taking those the ventral side of the 2-cell embryo and letting it grow. It'd basically be like that. Okay, so dorsalizing factors. And so what I just said, which is probably still true, you've got beta-catenin and veg-T here, and beta-catenin because of the dorsalizing factors, and veg-T, and that causes the production of CMWA, or however a French person might say it. And so CMWA is a transcription factor, and that's going to turn this new coop center into the, turn this into the new coop center. Now, and it's going to do this by upregulating XNRs. So you've got XNRs being produced all the way through here, but here, suddenly, boom, they go really high because of CMWA. Okay, now here's the frustrating part. I was talking about how now the chapter goes all the way back to early to mid 20th century, and the experiment showing that the new coop center is important for making an axis. So we take this 2 cells from this new coop center, put them into a different embryo on the opposite side, and each side of that embryo then forms its own axis. So that's how they originally figured out that this new coop center was important. I believe it was a guy named Newcoop who found that. Okay, so fate mapping, blah, blah, blah. I did go over this several times. So let's just do the experiments here. So this is kind of the control experiment for the ones that are coming up. So you take a blastula and you cut the top of it off. So you've got just epidermal ectoderm. You let it culture overnight. And the next day, all you've got is a ball of epidermal ectoderm, okay? So this is not being changed at all at this stage. We're gonna get rid of these marginal zones because they're complicated, we'll get back to them. If you take just your vegetal sections, you let them culture overnight, you get just endoderm. So at this point, there's no specification happening at either the top or the bottom of the embryo. Here's where all the action is happening. Okay, so now we're going to look at what's going on in these equatorial parts. So again, you take your top, you get ectoderm, you take your bottom, you get vegetal endoderm. Now these ones in the center, if you take the ventral side of it and you culture that overnight, you're going to get mesoderm, ventral mesoderm. Blood, when you see blood, think blood vessels, and heart. So if you look at your body, dorsal's back here, ventral's up here. This is where your heart is in the front. So this is ventral mesoderm. Now, if you take these marginal cells from the dorsal side and let them develop, you've got dorsal structures. You've got back muscle, you've got nodal cord, which becomes spinal cord, and you've got neural tube instead of ectoderm. Here, that little bit of ectoderm you have there becomes epidermis. So what this tells us then is that this blastula stage, you've already got some specification happening. The cells aren't what they're going to become. They're not what they're fated to become, but inside the cells, you've got transcription factors activated that are going to imprint on those cells and make them become something else. And that's done already. So I guess my point is, this isn't blood mesoderm yet. It's still just ectoderm and mesoderm, but it will become that at this point because the transcription factors have been turned on. I hope that wasn't more confusing. And here, another early experiment, and this is where you take those equatorial sections out and you put that ectodermal cap right on top of the endoderm. Now, you know, if you culture these separately, you'd get epidermis and endodermis. I just made that word up. Endoderm cells. You culture them together and you're getting induction of mesoderm. You're getting mesodermal structures coming up. So this is how they early on figured out that endoderm is releasing something that's diffusing to the ectoderm and causing the ectoderm to become mesoderm. Okay, early experiments. Here is what the signal is. Now, what's important about this? I've been talking about BMPs and XNRs forever. These are extremely important for inducing mesoderm, and they're both TGF-beta-like signals. So TGF-beta signaling is going to turn endoderm into ectoderm into mesoderm. But we've got two different signals, really, BMP4 and the XNRs. Very similar. If you had an actin type 2 receptor, which is this one here, it would affect both of these because it can cohabitate with them. But if you're just talking about what's actually going on in vivo, these BMP4s are going to activate SMADs 1, 5, and 8, and XNRs are going to activate SMADs 2 and 3. So this means these are transcription factors. So they're going to bind to different DNA sequences. So if you activate 2 and 3, it's going to bind to 4 and activate certain genes. If you bind to 1, if you activate 1, 5, and 8, they bind to 4 and they activate different genes. And you can see what the genes are doing down here. What another kind of neat thing to watch here is they both go through SMAD4. So these receptor SMADs are different, but the cytosolic SMAD is the same for both of them, which means, as we'll see shortly, if you have something that knocks out SMAD4, you knock out both BMP4 and XNRs. But in vivo, we've got two different things going on. Your BMP4s are producing mesoderm of the ventral variety, okay? And the XNRs are producing more dorsal mesoderm. And if they're high enough, we're gonna get Spearman organizer, and that will be our axis forming cells. Okay.
I have a quick question
Okay.
regarding the last slide. So say if we do perform an experiment and if we don't see a mesoderm, can we
Just hang on a second. Could you back up and go do that again? I missed it for a second.
so if say we cultured an experiment and we see an embryo without a mesoderm, so can we go on to hypothesize that maybe the SMAD4 receptor is missing? That's why we couldn't induce both XNRs and BMP4s.
Yeah, that's a good point. That's a good point. If you have, one of these mutated embryos that I always talk about where you get no mesoderm, it could be SMAD4 knocked out because then you're getting no TGF-beta-like signaling there. If I said you got only ventral mesoderm, then you would say, well, it must be SMADs 2 or 4.
3.
2 or 3.
Yeah.
But if it was like, oh, you don't get any ventral mesoderm, it would be the other SMADs. So that's kind of your 4 point there.
All right. Okay. Thank you.
Okay. Sure. So anyway, so we've got these diffusible factors coming from the endoderm that are causing the ectoderm to become mesoderm. Why don't we get all mesoderm and no ectoderm? We've got these two factors that are blocking the movement of those diffusible factors and limiting their reach. Okay, one is ectodermin. I'm not sure what I said in the last class, but I was wrong. Ectodermin is a ubiquitin ligase. So this is, again, through that ubiquitin pathway. And it is ubiquitinating these XNRs, ubiquitinating SMAD4 and blocking all of that mesoderm induction. This is actually, I said FOX11E. If you see it in a serif font, this is sans serif, and it looks like 1111. You see it in an actual serif font, which means the little capitals on the top and bottom. This is actually FOXI1E, so FOX11E. So I corrected that in your notes, but it's neither here nor there. Just think Fox, whatever E is this factor. So this factor, they don't explain why in your book, but it's stopping any of these cells from migrating up here and becoming, turning this into a mesoderm. So these two factors then are limiting the spread of mesoderm. So you get a band of mesoderm around the middle, but it doesn't continue on up to into the animal pole anywhere. And here, this is just a top view, just to remind you that, this is a three-dimensional structure. We always see it in 2 dimensions and cut through laterally. If you cut it across this way and look down on it, you've got a circle of mesoderm all the way around. And one end of this circle is going to be dorsal and the other ventral. And that's because, as we'll see in the next slides, this is releasing BMP4 blockers, which are diffusing this way and this way and this way.
I have a question.
Yep.
Based on this slide, right? So if something knocked out FOX11E, would there be no ectoderm layer and there would just be like mesoderm and endoderm?
I'm not sure the extent that FOX11E knockout would affect the embryo. It would work, but a lot of times there's some redundancy. I think if you knocked out ectodermin, then you would definitely get mesoderm coming up a lot higher. And I imagine if you knocked them both out, it would probably come almost up to the top. Okay, or maybe even all the way to the top.
Okay.
So I would say you would have to knock them both out for that experiment, but knocking either one out is going to affect it probably, and you'll get more mesoderm than you should. Okay, so this isn't that important. It's just basically saying somewhere between 4 and 11 hours after fertilization, you have to induce these cells, these ectodermal cells, to become mesoderm. If you wait till after that, they're never going to become mesoderm. They'll stay ectoderm. So if you try and induce them with activin or TGF-beta out here, they're going to stay ectoderm. But that doesn't mean they're mesoderm here. They've just been induced, which means some transcription factors are coming up. And later, those transcription factors will make them competent to become mesoderm. And that transcription factor, as we all know, is brachyury. Brachyury. Let's go forward to that and then come back. Our old friend brachyury. Okay, that is your mesodermal specifier. Okay. So this isn't that important. Don't worry about it. This is important. This is like, why is it you get to 12 divisions before you get any transcription from the zygotic nuclei? So I said, all the proteins made in the embryo up to the 12th division are made from maternally laid messenger RNAs. After that 12th division, then the nuclei get turned on, and that's when inductions really happen. Before that, you're preparing, but you're not really doing anything yet. Okay, and why is that? Probably, this is a hypothesis, but they think this is why. Early in the embryo, you've got one tiny little nucleus and a huge cell. So if you've got nuclear blocking factors, transcription factors that are repressing the nucleus, at this stage, you've got a lot of them compared to the amount of DNA there. As you go through round after round of mitosis without the cell growing any, you get to a point where there's a lot less cytoplasm per nucleus, which means you've kind of diluted out all the repressing factors. And now the nucleus starts to transcribe. I guess that's not what I wanted. Okay. So that is why you get zygotic transcription after the 12th division. And I think in zebrafish, it was after the 8th division, if I'm not incorrect. Okay, so now what they're showing through these early experiments is that even in this early blastula stage, this is dorsal and this is ventral. Because if you take these animal caps and you expose them to this, you will get dorsal mesodermal structures. And if you take animal cap cells and expose them to this, you get ventral mesodermal structures. Okay, and again, it's like what I find frustrating about this is they tell you this is dorsal and this is ventral because you've got these transcription factors there and these hormones, and that's what makes it dorsal ventral. Now suddenly we're going 100 years earlier. Is this dorsal? Is this ventral? How do we know? And it's like, Jesus Christ, guys, why don't you start with this and then go to the transcription factors? That's the way I'm going to do it next time. So you guys are still guinea pigs. Okay, so positional information, this is what they're learning there, that you've got your vegetal is releasing XNRs and BMP and a lot of XNRs here. That turns this into the organizer. Okay. And this is kind of bullshitty too. Don't worry about that. Did I just say that word? So here's how you make a dominant negative. Now, this is neat, and I may ask a question about this. If you take a receptor, an important, like a BMP receptor, an XNR receptor, or whatever, and you cut, uh-oh, my computer's going to die soon. If it does, don't leave. I'll be back when I get my wife's computer. Stupid computer. I'm waiting for parts to come in to fix it. Okay, so if you cut off the cytoplasmic domain, it no longer can transduce a signal. So you take messenger RNA for that, you inject it at the 2-cell stage, so you're overexpressing this. You're going to knock out all signal transduction through that hormone. And you can do this with any hormone. You can knock out, FGF, you can knock out XNRs. All right, I'm going. I'll be back. Computer dying.